I've always loved the ocean. The natural world has just always fascinated me.
A whale shark, South Passage.
Copy that, moving into your 12:31 o'clock.
I've always felt a strong sense of urgency to protect it.
Yeah, crew, get ready, master snorkels on.
Coming into your 12 o'clock landing boat.
Especially those lesser known species like fish and sharks. Whale sharks are iconic species. They're the biggest fish in the sea, and for 4 months of the year we're lucky because they come and visit us right here on the coast of Ningaloo. However, a lot of sharks and rays are ocean wanderers, and so after they leave these coastal environments, they're out into the blue. Environmental DNA offers a really powerful and a potentially really scalable way to track individuals and organisms as they move through their environments using only the genetic signature that they leave behind. Because we don't have to find the animals themselves. Instead, we can sample seawater and pick up those genetic signatures that they've left behind. Woo! I am stoked. It was an absolutely magic experience. Love the goods here. So now we can take it back to the lab.
We're here at Ningaloo Reef in Western Australia. It's a World Heritage Area that's got some amazing marine biodiversity. It's a marvellous place to study life on this earth. We're in a remote area of Western Australia. Doing science here is difficult. The Minderoo Foundation's Exmouth Research Laboratory is a place for scientists to run experiments and to deliver the best possible science for the region. So the Oceanomics program, embedded within the Minderoo Foundation, has a simple aim: develop better ways to listen to nature and make better decisions. We do this through the lens of genetics. Environmental DNA, or eDNA as it's known, refers to the fact that all animals, when they move through any environment, that's yourself or a fish in the ocean, is shedding small bits of genetic material, breadcrumbs if you like. The real power of environmental DNA lies in its ability to look at the entire tree of life. So while traditionally we've gone out and counted fish or corals, there's a myriad of other species, from bacteria to microeukaryotes to all the things nocturnal fish that are not easy to count. They shouldn't be ignored. If we look at the ecosystem in its entirety, rather than count a few things that are easy to see, we can make better decisions. You might have heard of the IUCN Red List that assess whether an animal is threatened, endangered, critically endangered, or extinct. eDNA data can be directly ported into the IUCN's Red List assessment. That is work that the Minderoo Foundation and IUCN are partnering on as we try and figure out where animals are, how they're moving, and indeed whether their threat status should be elevated or decreased. Figuring out where animals live and how they're trending is vital ecosystem intelligence.
I'm Abby. My specialty is in environmental DNA. My research specializes in whale sharks. Almost 30% of shark species globally are threatened by extinction. Their decline can have significant effects on the marine ecosystem. During the last 75 years, whale shark populations have faced a significant decline. And due to this, in the year 2016, they were listed endangered by the IUCN Red List. A lot of the questions about the biology of these whale sharks that are important to inform conservation is unknown to us. Traditionally, genetic sampling of whale sharks is done through tissue biopsies. With non-invasive eDNA technique, we can get the genetic signature of the animal by just collecting seawater in its vicinity. The seawater sample that we collected right behind the whale shark individuals, when we genotype them, we can see that it matches really closely to the genotype data we got from the same individual shark. This can help us avoid invasively genetically sampling this individual. Now that we can identify whale shark individuals from just a seawater sample, we can inform about the population genetics monitor this species effectively and better inform conservation, even without actually seeing them.
So what we see here is about 6,500 environmental DNA samples, and we've found over 70 threatened species. What we hope in the future is we ramp up the amount of sampling through community engagement and more samples that researchers take. We're able to start to build temporal and spatial maps of where threatened species go and the areas we should protect. To give one example of the power of environmental DNA, a single 1 litre water sample from here at Ningaloo Reef will yield around 100 species of fish, not to mention the corals, the sponges, all the other biology. When we amplify that on a global scale, and we do tens of thousands of samples, we start to get powerful oceanic scale data sets, enable us to make better decisions here and elsewhere around the globe.
I'm really excited about the scalability of eDNA. We can use DNA signatures to study the entire tree of life from a simple water sample. We can't manage what we don't understand. As we decode the ocean's secrets, even through invisible traces of life, these new tools are opening up fresh windows, giving us the knowledge that we need to help protect natural ecosystems before time runs out.